Okay, so in this lecture, what we're going to get into is the nucleic acids, and this will be sort of a short segment on the macromolecules and specifically nucleic acids and proteins. We will discuss the lipids a little bit when we get to plasma membrane and other membrane ideas at the cell level. So I don't want to talk about them now. And then, of course, carbohydrates really are not going to play a huge role in processes related to development, so we won't spend a whole lot of time on those either. So just a reminder about the macromolecules. Remember that they are large, they have a polymeric structure to them, they're built out of some kind of a subunit, so that'll definitely become sort of important here. And I think what you need to really kind of be focused on in these next two lectures is the idea that as we look at their structure and what they look like, that is largely going to determine what kinds of functions they're going to involve in, or more specifically, when we think about their functions, their structure is going to determine how they go about doing those functions. So form and function do go together, and as we think about the functions as they show up throughout the course, you just need to remember that this is what the molecules look like and why certain ways that we do things as a cell are tied to that particular restriction. So in this lecture, we'll focus primarily on the nucleic acids, and what we'll do is we'll talk about DNA in a little bit more detail, and then just for RNA, we'll talk about some of the differences with RNA without going through the structure of RNA in as much detail. So certainly the name tells us what it is. It's a deoxyribonucleic acid. Acid tells us a little bit about its nature. And in fact, the deoxyribo also tells us a little bit about its nature. As a macromolecule, it is made out of subunits, in this case called nucleotides. We've talked a little bit about these as examples from time to time throughout the previous lectures. I suspect all this is stuff that you've seen before. We've got the 4 different cyclic bases, G's, A's, T's, and C's. Remember I told you that these macromolecules are held together by covalent bonds, and in this case the specific covalent bond that holds the subunits together is what we call a phosphodiester bond. So if you remember your organic, an ester is some kind of a bond across an O, and this would be 2 ester linkages with a phosphate in the middle. That's basically all that means. And what's a little bit more important is when you think about the overall structure of the DNA, the phosphate group of one is going to interact with the OH group of another. This is talking about the sugars, and this is what's going to give some orientation to that molecule with regards to the formation of that phosphodiester bond. It is going to play an important role in the way in which we build nucleic acids and the way we use nucleic acids in the processes that they are important for, mostly the genetic information, and not just the storage of the genetic information, but passing along that genetic information and also expressing that genetic information. So you guys will probably remember the 5 and 3' ends, and all of this is caused by the orientation of that phosphodiester bond. So let's look at a couple of things here. Introduced these early in the chapter where we looked at the hydrogen bonding. Well, now we can look at the details a little bit on these building blocks. Deoxyribo refers to the ribose, and the nucleo refers to the base. And we have 4 different bases here, the G, A, T, and C. And of course, A's and T's hydrogen bond with each other, and G's and C's hydrogen bond with each other. We'll talk about that secondary and tertiary structure here in a second. These nucleotides also have phosphates attached, and anywhere from 1 to 3, and it'll be this phosphate that's going to interact with the 3' OH of the next nucleotide. So when you put these things in a chain, you can definitely see some of those emergent properties coming out, where you've got the 5' has a phosphate at the end, the 3' has your sugar at the end, specifically that OH and that 3. So basically these numbers is 1, 2, 3, 4, 5 carbons in a ring. So 5' refers to the fact that the 5th carbon is showing, and 3' refers to the fact that the 3rd carbon is showing. Here's your phosphodiester bond with one ester linkage, two ester linkages, and a phosphate in the middle. And so notice that the backbone structure is common. If we were to just kind of blank out all of these bases, you wouldn't be able to tell any difference between these subunits. It would be just one normal carbohydrate chain held together by these phosphodiester bonds. It is the base that actually gives each individual nucleotide its identity and separates the G, A, T, and C from each other. So this is what one side of a chain would look like. Obviously, with regards to DNA, we know it's a double helix shape. So you'd have another chain on this side interacting through those base pairs. And we won't go through all the stuff that went into Watson and Crick trying to figure that out. That's more for a genetics course, but just to summarize a few things, once they form that double helix through those complementary base pairs, then what we end up with, and remember base pairs are A pairing with T for 2 hydrogen bonds, 3 pairing with C for 3 hydrogen bonds. We had that in that previous discussion, talking about complementarity and lock and key fit. So definitely tie this back into that conversation. And in order for the complementarity to show up in this case, the two strands actually have to be anti-parallel to each other. That's probably a term that you'll remember from maybe even high school biology. And so that's just referring to the 5' and 3' ends of the two chains. And let's just bounce forward to this picture here. You can really see it. So here's your 3 hydrogen bonds between G's and C's. Here's 2 hydrogen bonds between A's and T's. And notice that your sugars on the two sides are upside down relative to each other. And that's because this one's going 5 to 3. This one's going 5 to 3 the other way. So that's what anti-parallel means. The DNA inside of a cell is found in individual molecules that we call chromosomes. So those don't really matter when it comes to expressing. Chromosomes are important for the storage and safety of the genetic material, and as we'll see, very important for cell division and making sure that every cell that comes from cell division actually gets a copy of that information. So here's what chromosomes might look like. These very specifically are chromosomes in a cell that is dividing. Because remember, when we go to divide, we're going to have duplication, so DNA replication is taking place. We'll talk about it in more detail. And these chromosomes for sure have been actually compacted down during prophase in order to be able to separate. So our chromosomes in our normal cells don't really look like this. They would be much less packed up, and typically when you see a chromosome like this means that its genes are no longer active. And remember, I told you that the division of a cell takes about an hour, and this is why, is because when you're packed up this tight, you can't express your genes. So we can't be tied up in mitosis or meiosis for very long. So this is what the chromosomes look like for us as humans. We have the 23 pairs, and you can see that their formats may be a little bit different. The location of the centromere. We'll get into some aspects of how that works when we get to the cell division. So that's just kind of the basics of DNA and their structure, or its structure. Let's think a little bit about the RNA and how it's different. This is probably stuff you're aware of as well, where RNA nucleotides, instead of deoxyribose, they have ribose. And of course what that means is that the ribose is going to have an extra sugar, hence the deoxy. And it has it's not the 3' sugar, it's the 2' sugar, or 2' carbon of the sugar. It contains uracil instead of thymine, even though the two are very similar to each other. In fact, they're similar enough to where thymine pairs with A in DNA, uracil pairs with A in RNA. And of course, one of the other big differences has to do with its stability and its what we might call its lifespan, how long it stays around. RNA molecules tend to be fairly short-lived, and we use them in that way. And one of the reasons why is because RNA molecules tend to not form double helices. They tend to exist simply as single strands. So when you think of things like a messenger RNA or a transfer RNA or any of the others that are found in a cell, generally those things are single-stranded, and they would have slightly less stability and not live quite as long as their DNA counterparts. Another contributor to that is the ribose. The lack of this oxygen. Oxygen is very reactive, so the lack of this oxygen on DNA also gives DNA a little more stability that way as well. So RNA, even though it is less stable, it still fulfills the jobs we wanted to do. And in fact, we want it to be less stable because the jobs we wanted to do are very temporary. We don't want them to be around for very long. So it's not a bad thing. Obviously, this is something that we want our RNAs to have in place. So we'll talk about the way in which nucleic acids get passed on for the cell division, and we'll talk about gene expression, their roles in gene expression as well. All right, so next time we'll talk about the proteins and then get into some cellular stuff. Bye.